Vitamin K2 is a group name for a family of related compounds, generally subdivided into short-chain menaquinones and the long-chain menaquinones, of which Mk7, Mk8 and Mk9 are nutritionally the most recognized. Forms. All K vitamins are similar in structure, they share AA-O equinonia ring, but differ in the length and degree of saturation of the carbon tail and the number of O side chains SA. The number of side chains is indicated in the name of the particular menaquinone, and this influences the transport to different target tissues. Mechanism of action. The mechanism of action of vitamin K2 is similar to vitamin K1. Traditionally, K vitamins were recognized as the factor required for coagulation, but the functions performed by this vitamin group were revealed to be much more complex. K vitamins play an essential role as cofactor for the enzyme I-cubed glutamyl carboxylase, which is involved in vitamin K-dependent carboxylation of the GLA domain in GLA proteins to I-cubed carboxyglutamic acid and these proteins. Carboxylation of these vitamin K-dependent GLA proteins, besides being essential for the function of the protein, is also an important vitamin recovery mechanism since it serves as a recycling pathway to recover vitamin K from its oxide metabolite for reuse in carboxylation. Several human GLA-containing proteins synthesized in several different types of tissues have been discovered, coagulation factors, as well as anticoagulation proteins. These GLA proteins are synthesized in the liver and play an important role in blood homeostasis. Osteocalcin. This non-collagenous protein is secreted by osteoblasts and plays an essential role in the formation of mineral in bone. Matrix GLA protein. This calcification inhibitory protein is found in numerous body tissues, but its role is most pronounced in cartilage and in arterial vessel walls. Growth arrest specific protein 6. GAS6 is secreted by leukocytes and endothelial cells in response to injury and helps in cell survival, proliferation, migration, and adhesion. Proline-rich GLA proteins, transmembrane GLA proteins, GLA-rich protein and periostin, whose precise functions are still unexplored. Health effects. Bone density equals, vitamin K2 deficiency results in a decreased level of active osteocalcin, which in turn increases the risk for fragile bones. Research also showed that vitamin K2, but not K1 in combination with calcium and vitamin D can decrease bone turnover and that vitamin K2 is essential for the maintenance of bone strength in postmenopausal women. The Japanese population seems to be at lower risk for bone fractures compared to European and American citizens. This finding would be paradoxical, if levels of calcium consumption were the only factor determining bone density. Studies linked Japan's greater levels of BMD to that country's widespread consumption of natto. Increased intake of Mk7 from natto seems to result in higher levels of activated osteocalcin and a significant reduction in fracture risk. Heart calcification patients suffering from osteoporosis were shown to have extensive calcium plaques, which impaired blood flow in the arteries. This simultaneous excess of calcium in one part of the body, and lack in another which may occur even in spite of calcium supplementation is known as the calcium paradox. The underlying reason is vitamin K2 deficiency, which leads to significant impairment in biological function of MGP, the most potent inhibitor of vascular calcification presently known. Fortunately, animal research showed that vascular calcification might not only be prevented, but even reversed by increasing the daily intake of vitamin K2. The strongly protective effect of K2 and not vitamin K1 on cardiovascular health was confirmed by, among others, Geliage et al. In the Rotterdam study performed on a group of 4,800 subjects. Results of more than 10 years of follow-up were verified, also by Gast et al., who demonstrated that among K vitamins, the long-chain types of K2 are the most important for efficiently preventing excessive calcium accumulation in the arteries. Absorption profile of different K vitamins. Vitamin K is absorbed along with dietary fat from the small intestine and transported by chylomicrons in the circulation. Most of vitamin K1 is carried by triacylglycerol-rich lipoproteins and rapidly cleared by the liver. only a small amount is released into the circulation and carried by LDL and HDL. MK4 is carried by the same lipoproteins and cleared fast as well. The long-chain menaquinones are absorbed in the same way as vitamin K1 and MK4, but are efficiently redistributed by the liver in predominantly LDL. Since LDL has a long half-life in the circulation, these menaquinones can circulate for extended times, resulting in higher bioavailability for extrahepatic tissues as compared to vitamin K1 and MK4. Accumulation of vitamin K in extrahepatic tissues has direct relevance to vitamin K functions not related to hemostasis. Dietary sources and adequate intake. In 2012, Canadian health writer Kate Copyright Orme suggested the recommended daily allowance for K vitamins might be too low. Earlier suggestions in the scientific literature, which note that the RDA is based on hepatic requirements only, date back as far as 1998. This hypothesis is supported by the fact that the majority of the Western population exhibits a substantial fraction of undercarboxylated extrahepatic proteins. Thus, complete activation of coagulation factors is satisfied, but there do unregistered trademark Ts seem to be enough vitamin K2 for the carboxylation of osteocalcin in bone and MGP in the vascular system. Highest concentrations of vitamin K1 are found in green leafy vegetables, but significant concentrations are also present in non-leafy green vegetables, several vegetable oils, fruits, grains and dairy. In Europe and the USA 60%, or more, of total vitamin K1 intake is provided by vegetables, the majority by green leafy vegetables. National surveys reveal that K1 intakes vary widely. Intakes determined by weight. Dietary intakes are similar in mainland Britain to the USA, with average daily intakes of around 70 €80 1/4 g, which is less than the adequate intake for vitamin K. Apart from animal livers, the richest dietary source of long-chain menaquinones are fermented foods, typically represented by cheeses in Western diets and natto in Japan. Food frequency questionnaire-derived estimates of relative intakes in the Netherlands suggest that 90% of total vitamin K intakes are provided by K1, 7.5% by MK5 through to MK9 and 2.5% by MK4. Most food assays measure only fully unsaturated menaquinones. Accordingly, cheeses have been found to contain MK8 at 10 €20 1/4 g/100 g and MK9 at 35 €55 1/4 g/100 g. Dietary intake sources. Vitamin K2 is preferred by the extrahepatic tissues, and this may be produced as MK4 by the animal from K1, or may be of bacterial origin. The latter may be consumed already prepared by bacteria. Discussion is ongoing as to what extent K2 produced by intestinal bacteria contributes to daily vitamin K2 needs. If, however, intestinal bacterial supply was enough to supplement all tissues needing K2, we would not find high fractions of undercarboxylated GLAP proteins in human studies. Natural K2 is also found in bacterial fermented foods, like mature cheeses and curd. The MK4 form of K2 is often found in relatively small quantities in meat and eggs. The richest source of natural K2 is the traditional Japanese dish natto made of fermented soybeans and Bacillus subtilis, which provides an unusually rich source of K2 as long-chain MK7. Its consumption in northern Japan has been linked to significant improvement in K vitamin status and bone health in many studies. The intense smell and strong taste, however, make this soy food a less attractive source of K2 for Westerners' tastes. Supplement food companies sell natto extract, standardized for K2 content, in capsules. It is not known whether B. subtilis will produce K2 with other legumes. Anticoagulants and K2 supplementation. Recent studies found a clear association between long-term anticoagulant treatment and reduced bone quality due to reduction of active osteocalcin. OAC might lead to an increased incidence of fractures, reduced bone mineral density, bone mineral content, osteopenia, and increased serum levels of undercarboxylated osteocalcin. Bone mineral density was significantly lower in stroke patients with long-term warfarin treatment compared to untreated patients, and osteopenia was probably an effect of warfarin interference with vitamin K recycling. Furthermore, OAC is often linked to an undesired soft tissue calcification in both children and adults. This process has been shown to be dependent upon the action of K vitamins. Vitamin K deficiency results in undercarboxylation of MGP. Vascular calcification was shown to appear in warfarin-treated experimental animals within 2 weeks. Also in humans on OAC treatment, twofold more arterial calcification was found as compared to patients not receiving vitamin K antagonists. Among consequences of anticoagulant treatment, increased aortic wall stiffness, coronary insufficiency, ischemia, and even heart failure. Arterial calcification might also contribute to systolic hypertension and ventricular hypertrophy. Coumarins, by interfering with vitamin K metabolism, might also lead to an excessive calcification of cartilage and trachobronchial arteries. Anticoagulant therapy is usually instituted to avoid life-threatening diseases, and a high vitamin K intake interferes with the anticoagulant effect. Patients on warfarin treatment, or treatment with other vitamin K antagonist drugs, are therefore advised not to consume diets rich in K vitamins. However, the latest research proposed to combine vitamins K with OAC to stabilize the INR. Toxicity: there is no known toxicity associated with high doses of menaquinones. Individuals taking anticoagulant medications, such as warfarin, should consult their doctor before taking vitamin K2. Unlike the other fat-soluble vitamins, vitamin K is not stored in any significant quantity in the liver. Therefore, toxic level is not a described problem. All data available at this time demonstrate that vitamin K has no adverse effects in healthy subjects. The recommendations for the daily intake of vitamin K, as issued recently by the Institute of Medicine, also acknowledge the wide safety margin of vitamin K. A OEA search of the literature revealed no evidence of toxicity associated with the intake of either K1 or K2. A point of concern is, however, the potential interference of K vitamins with OAC treatment. Individuals taking anticoagulant medications, such as warfarin, should consult their doctor before taking vitamin K2. Animal models involving rats, if generalizable to humans, show that MK7 is well tolerated. References